Hello, thank you for the very kind invitation to present at the NC UK Virtual Summit. I'm Chrissie Thirlwell. I'm a clinical academic, a medical oncologist that looks after patients with neuroendocrine tumours for 1 day a week, and the rest of the time I do research at the University of Exeter in neuroendocrine tumours, cancer genomics, and I've also been involved in the 100,000 Genomes project. Over the next 25 minutes or so, I'm going to talk about some of the genomic advances in neuroendocrine cancer research. I'm going to start with a bit of a genomics 101 so that we're all starting from the same page. And the human genome is a wonderful thing. There are 3 billion base pairs of genetic code that measures over 1 meter that packs into a cell nucleus just 6 microns across. And every time a cell divides and multiplies, that sequence is unraveled, it's copied and then packaged back into the cell nucleus. It's an absolutely phenomenal undertaking. Some of you may remember the Human Genome Project, and rather alarmingly, this was completed nearly 20 years ago now, and it was a massive international research collaboration which took over 10 years from 1990 to 2000 and cost nearly $3 billion US. And that work created the first roadmap, the first sequence of the human genome. And researchers and scientists now use this roadmap every day in their research. There have been so many advances in terms of understanding cancer and learning about which drugs to use to treat cancer that have been related to and use this roadmap every day in research. I'm just going to tell you a little bit more about how the genome is packaged up and unpackaged, because that's quite important when we're learning about neuroendocrine cancer genomics. So in the middle here, you can see there's the code. So there's the different letters of the genetic code. There's 3 billion of these. And then that's packaged up using these chromatin remodelers in the middle of the screen there. That's important because some of these genes are mutated in neuroendocrine tumours, and then it's packaged up further into the chromosomes. So each cell has pairs of 23 chromosomes, and every time the cell divides, it's unwrapped, copied, and put back together again. Alongside the letters, the sequence, there is methylation. So these are little methyl lollipops which are tagged along the genome, which can turn genes on and off. There are histone modifications, and these are attached to these building blocks that the sequence is wrapped around to condense it into the chromosomes. And there's also microRNAs, which is another way that genes can be turned on and off without affecting the DNA sequence. So the DNA sequence in our chromosomes encodes genes, and these are building blocks which make proteins, which build everything about our body and how it works. And we think there are about 30,000 genes. So we have these 6 billion letters, but only 1% of those is actually used to code for these genes, which make our protein building blocks. It's important to understand the difference of if a mutation occurs in what we call the germline or in the somatic cells of the body. So on the left-hand side is a diagram showing what might happen if there's a germline mutation. And that's a mutation in the DNA we might inherit from our mother or our father. So at the top of the left-hand picture, there's an egg and a sperm. So the egg has 1 copy of all the mother's DNA information, and the sperm has the father's copy. If there's a mutation in one of those when the baby's formed, then that's inherited, and that mutation is present in every cell of the body, and then that person can pass that mutation on to their children. There are different ways that these can be inherited, but it's important to understand the difference between an inherited mutation, because there are some neuroendocrine cancers which are related to this, and a somatic mutation. On the right-hand side, you can see in the purple, the variant in this situation is not in the egg or the sperm that's inherited from the parents. These mutations occur in the cell either when the baby's developing, in youth when you're growing up, or when you're an adult. And there are various things in our lives that can affect and damage the DNA in our cells which haven't been inherited in the germline. For example, smoking can damage cells, in relation to lung cancer, and UV light can damage cells in relation to melanoma. So that's the difference between germline and somatic variants, because I'll be talking about those later on. So I've been working in cancer genomic research for over 15 years now. And the rate that the field has changed has been absolutely phenomenal. I did my PhD in 2004, and at that time I was working in bowel cancer, and it took me nearly 6 months to sequence one gene, a big gene related to bowel cancer. And now we can send off a DNA sample and we can get the sequence of all the genes back, all of the 30,000 genes, in about 48 hours. So the pace at which we create this information has improved phenomenally. And the technology alongside how we extract the DNA and sequence the DNA has just been expanding exponentially. So there's two examples here on this slide. On the left, this is a scientific journal, so Science Translational Medicine, and there is a publication and a paper in this which really blew me away. It's quite a few years ago now, but what they managed to do in this publication is they took a blood sample from a mother, so an expectant mother, and in that blood sample you can extract DNA from the baby that's circulating around in the bloodstream, and you can look for things such as Down syndrome. So you can do a non-invasive blood test to look for Down syndrome. And the technology of finding that circulating DNA in a blood sample, where there'll be the mother's DNA as well, is absolutely astounding. And we've used the similar approach to look for circulating DNA in cancer patients as well. And I've got an example of that later on in the presentation. And on the right here, this again was from about 10 years ago now. I was sat having my breakfast one Sunday morning, and Ozzy Osbourne decided at that time to spend $10,000 US to have his genome sequenced. And at that point, it's what we call the exome. So that's the 1% of the coding genome. He spent $10,000 doing that. It now costs us a few £100s to do this. And he thought it would be a good idea to share the findings with everyone in the Sunday Times magazine. And I was there chuckling to myself, thinking, oh, maybe they identified the worshipping the dark lord gene or the biting the head off chickens genes. But actually, in a very serious way, we have to be very thoughtful about this information. It's deeply, deeply personal information. You may have read earlier this year that Ozzy's been recently diagnosed with Parkinson's disease. And how do you think he might have felt if 10 years ago they'd identified some changes which would tell him he might develop Parkinson's disease in 10 years, but we don't really know when that might happen? So how do you think he might have lived his life? And how do you think you or I might have lived their lives? So it's deeply, deeply personal information. There's an enormous amount we can learn from studying the cancer genome. So what we can do, we take the tumour itself and we extract DNA from the tumour. It's important to also extract normal DNA from normal tissue as well. So we've got that individual, that person's roadmap, and then we can compare what's different in the tumour. So these are somatic changes in the tumour. But by studying the DNA and the genetic changes, we can learn about the biology, how the tumour develops, we can identify new drug targets, we can see how tumours evolve, we can see how they might develop resistance to the treatments we give them, and we can understand how tumours can unfortunately spread from where they originate, the primary tumour, and they can sometimes metastasize to lymph nodes or to other organs such as the liver. So there's an enormous amount we can learn by studying the DNA makeup of tumours. The first time that it was observed that there was DNA and there was changes that might be present in cancer cells was just at the turn of the century, in the early 1900s. And then since then, there have been some step changes in our understanding. Some of you may be familiar with Watson and Crick, understanding the double helix of DNA in 1953. And then following on from this, the first human genome sequence was in 2000. And to the right side of the slide, you can see there's been an enormous pace of change over the last 20 years or so. And bearing in mind that this paper was published at the 10th anniversary of the Human Genome Project, completing in 2011, at that point, they estimated that cancer genomic sequencing would be part of a routine diagnostic in 2020, and they were absolutely right, because it's something that's involved in how we look after cancer patients on a day-to-day basis now. So I was involved in the 100,000 Genomes project, and I'll say a little bit more about that in relation to neuroendocrine tumours later in the presentation. But this was a phenomenal undertaking where 17,000 cases of cancer from patients across England and the rest of the UK were sequenced. They underwent whole genome sequencing, so that's the whole 3 billion base pairs of the DNA sequence. And what they found in looking at 17,000 cancers is around half of them had a mutation in them, which could give us an insight as to what would be the best treatment for them. So either a new drug or potentially a clinical trial. But this is from across all cancer, so all of the common cancers: breast cancer, lung cancer, and bowel cancer. And something else that the Human Genome Project and understanding the DNA sequence has taught us is that we can now shorten the interval between identifying a target that we can give a medicine for, a drug for, in cancer therapy. So this slide just gives an overview of some of the discoveries that have been made going back since the 1960s. And so at the top there's a target of BCR-ABL, which identified in 1960. And on the right, you can see that it took till 2001 before we identified a treatment. And as you go through the slide, you go further down the slide, that the time between identifying the target and actually finding a drug which treats that target has shortened. So right at the bottom here, we call this ALK, ALK. This is present in lung cancer. So it's identified in 2007. And then in 2010, so only 3 years, we've gone from a 40-year gap to 3-year gap, a drug was given. And not only that, some of these cancers then develop resistance to those drugs. And within another 18 months, then I worked out where that resistance was and found a new drug to treat that. So this is in some of the more common cancers. You can see there's lung cancer there and also melanoma and a type of lymphoma. So what do we know about the neuroendocrine tumour genome? We know that clinically these are heterogeneous tumours that can occur in various sites in the body, most commonly in the small intestine and the pancreas and the lung. There are also some rarer neuroendocrine tumours that occur in the thyroid, the adrenal, and also some of the reproductive organs, so the ovary. Something that's very striking when we look at the DNA sequence of neuroendocrine tumours is what we call the background mutation rate is very low. So there are mutations in these somatic cells, so in the tumour themselves. Some of these mutations are what we call drivers. So it's a mutation in a gene that's key to actually making the cancer develop and grow. And some of these mutations are called passengers. So they're just there, and it may be a byproduct of something in the environment. So again, an example here is lung cancer, where smoking can damage the DNA, or melanoma, where UV light can damage the DNA. But the arrow at the bottom of the figure here on the right is the background mutation rate for neuroendocrine tumours. And it's very, very low. It's lower than virtually any other cancer type that's been sequenced. And I think that tells us something quite important about these tumours, and we're still researching this. We don't know exactly why this background mutation rate is very low, but it's something that's very striking in the tumour type. This slide actually puts together over 10 years efforts of sequencing research in neuroendocrine tumours. So I was involved in the project looking at the small intestinal NENs. We called them SI NENs in the top here. We had to sequence over 180 tumours to identify whether there were any mutations whatsoever. And there are actually very, very few mutations. So the small intestinal NETs have a very low background mutation rate and overall very, very few mutations. So only 8% of cases have mutation in this gene, CDKN1B. What we've learned since is those tumours which have this mutation don't really behave any differently to the tumours that don't have this mutation. So as a cancer doctor, it's not really telling me much more about how that tumour develops. But something that my group's been working on over the last 5 years or so is we've been looking at some of the epigenetic changes to learn more about the the small intestinal nets. I don't have time to talk about that today, but there's ongoing research to really learn more about the biology of the small intestinal tumours. When we think about pancreatic tumours, I found that around 40% of cases have mutations in these chromatin remodelers. So if you remember the slide early on where I was discussing how the DNA gets wound around those building blocks, these are the chromatin remodelers, and we call that the epigenetic machinery. And again, in the pancreatic tumours, a number of germline mutations have been identified. So these are mutations which you might inherit from your mother or father. The other way in which germline mutations occur is it can just be just by error. So you might be the first person in your family where this mutation has occurred, but once it's there, there's a potential that you then pass it on to your children. So a number of germline mutations have been identified. And again, in bronchial neuroendocrine tumours, again, 40% of cases have these mutations in the epigenetic machinery, so those building blocks that the DNA is wound around. What's really striking to us as cancer doctors is, unfortunately, there aren't mutations in genes which we can then easily target with drugs. So if we go back to the previous slide here, there's a target that's identified, and then there are the drugs which fit to that target. And unfortunately, we don't really have these actionable mutations where we can go into the cupboard and get out the next targeted drug for that. But there's still an enormous amount of work going on in pancreatic and bronchial neuroendocrine tumours. And again, my group have been looking at some of the epigenetic changes, so some of the DNA methylation and other changes in those tumour types, just so we can learn more about how these tumours develop. And this slide just gives a very brief overview of some of the germline mutations that occur in neuroendocrine neoplasms. So there are inherited syndromes that you may have heard of. So there's multiple endocrine neoplasia, there's von Hippel-Lindau and neurofibromatosis, and around 30% of pheochromocytoma and paraganglioma cases, there might be a germline mutation, and there's quite a long list of genes that are involved in those tumour types. And most recently, we found that in pancreatic neuroendocrine tumours, there are up to 17% of cases there might be a germline mutation. So when we're seeing patients in clinic, we're obviously very thoughtful about whether one of these syndromes or whether a germline mutation might be relevant. And some of you may have been referred to geneticists, so clinical geneticists, to discuss this and perhaps have some tests done to see if that's the case. I'm just going to touch on an area of research that's still evolving, both in routine cancer care, but also in neuroendocrine tumours. Something that I mentioned earlier is that we can actually identify and pick up DNA from tumours that's circulating in our bloodstream. And alongside that, there's also circulating tumour cells, which can be identified. And this may be part of how tumours spread from the primary tumour off to other organs. So the circulating tumour cells can be identified in a blood sample, and alongside that, circulating tumour DNA can also be identified. And it's very, very, very challenging research because we have lots of normal DNA circulating in our blood from cells which are just popping and releasing their DNA. And it's like finding a tiny needle in a haystack to identify the tumour DNA and then sequence that. But there has been some research which has done this really nicely in other cancer types, where you can follow the mutations in the circulating tumour DNA, and you can see whether a patient's responding to their treatments. This is chemotherapy drugs. There's epirubicin and paclitaxel on this figure here. And on the left-hand side of the graph, you can see all the different coloured lines, and that's the level of the different mutations in the blood when they start their chemotherapy. And you can see that these all drop down during the chemotherapy. Unfortunately, it then starts to climb up again, just as the patient requires some more chemotherapy with paclitaxel. And what we know and what we've learned from this type of approach is we can sometimes learn whether someone's responding to their treatment before we might see any changes on the scans that we do. So you may all be very familiar with having different types of scans, CT scans and MRI scans, to see how you might be responding to treatment. And we have performed a small study, what we call a pilot study, looking for circulating tumour DNA in pancreatic neuroendocrine tumours. This was a few years ago, and there's some ongoing research in this area. There's been quite a large body of work as well from UCL looking at circulating tumour cells in different types of neuroendocrine tumours, but this is circulating tumour DNA, where what we did was we took some of the tumour, so these are somatic mutations. We identified what mutations were there in the tumour, and then we actively looked for them in the blood sample from the patients. And we identified that accurately, and we could see that some of the levels changed as the patients went through their follow-up. So I'd watch this space from that perspective. There's still ongoing work here, but there is quite a large body of work in looking at and using circulating tumour DNA in following other cancer patients. I'm just going to spend the next few minutes talking about the 100,000 Genomes project, because there was a neuroendocrine tumour arm of this in terms of research which is ongoing. Some of you may be aware of the 100,000 Genomes project. It completed in 2018 and it was launched in 2013 and 14, and it was a massive undertaking within the NHS and Genomics England, where over 97,000 patients across the UK were recruited. They focused on sequencing cancer and also individuals with rare disease, and they sequenced and created the whole genome. So that's 3 billion base pairs, 100,000 genomes, 3 billion base pairs, and that created over 21 petabytes of data. And just to put that into something a bit more realistic, 1 petabyte of music would take 2,000 years to play on an MP3 player. So that's an enormous amount of data, and it was an absolutely massive undertaking. There are over 5,000 staff, but something I'll be talking about is there are over 3,000 researchers now using this DNA sequencing for us to learn a lot more about cancer. And what they found, so they took over 122,000 samples and 118,000 genomes were created. Of those, 32,000 were cancer, so that's from 17,000 cases, because you need to do the sequence of the normal DNA and then the sequence of the tumour, and then 83,000 in rare disease. And something that was really striking is they found that in 50% of the cancer cases, there was a potential therapeutic target or a clinical trial that could be identified. And it's something moving forward that we're now going to be including in the day-to-day management of how we look after cancer patients, because it's so important for us to select the right treatment based on some of the genetic changes, so that we can target the tumour appropriately, give our patient the right therapy, and then if their cancer develops or changes further in the future, we sometimes might then take another piece of tissue and sequence that again to see what new mutations are there, because then that guides the next line of treatment that we might use. Something else that we been studying through the 100,000 genomes is something called pharmacogenomics. So all of our genetic makeup that we inherit from our mother and our father can actually affect how we might metabolize or handle different medicines. And this is on an individual basis. So I'm just going to go through one example of pharmacogenomics in chemotherapy, and it's a chemotherapy drug that we use quite regularly in neuroendocrine tumors. But we know that pharmacogenomics can affect how drugs are activated. So if you either have an injection or you take a tablet, pharmacogenomics can affect how quickly the drug is activated. It can affect how quickly the drug is deactivated. So if you have slow deactivation, it means more of the drug is left in the body for longer and you can have more side effects. And then some people have slower enzymes for this. And everyone inherits different variations in these enzymes. And these enzymes are the protein building blocks that I was talking about, that's coded by genes. So everyone's different, and we all actually manage and handle our medicines quite differently. And an example in neuroendocrine tumours is that with a chemotherapy drug of 5-fluorouracil. There's a tablet form of this called capecitabine. And capecitabine is used quite commonly in some neuroendocrine tumour types. And 7% of the European population have variants in this gene called DPYD, and we do look for this in patients that are starting their treatment. It's now being rolled out nationally, so not every single person is tested for this before they start their treatment, but it's something that's being rolled out across the country at the moment, because we know that if you have these variants or changes in this DPYD gene, it affects how you handle your chemotherapy. And if you break the chemotherapy down more slowly, you can unfortunately get more of the side effects and toxicities. So we can screen for this DPYD change, and then we can advise on the appropriate dose of the medicine. And some people, it's actually advised that you don't have that type of medicine because it can be really quite toxic. And alongside this, in the 100,000 Genomes project, we introduced Genomic Tumour Advisory Boards, and that's where all of the cancer cases were discussed, but we incorporated the genomic information, the sequencing information, and that then we discussed what might be the best treatment for that patient, or what might be a good treatment for them in the future. And this is something that we're planning and hoping to put together for neuroendocrine tumours moving forward. It's going to take a good few years to get into place, but it's something that we'll then be incorporating with our normal tumour board meetings to discuss any of the DNA changes in neuroendocrine tumours. Alongside that activity, there's also Genomics England Clinical Interpretation Partnership, shortened to GeCIP, and I'm the lead for the Neuroendocrine Tumour GeCIP, and we have a number of members from across different centres in the UK with a research background and clinical background. And the idea of these GeCIPs in the 100,000 Genomes project is to look at the research element of those tumours that were sequenced. In the neuroendocrine tumour GeCIP, we have tumours that have taken from a number of different sites. So we have some bronchial, intestinal and pancreatic tumours. So again, it's a heterogeneous group of tumours that we have included in our GeCIP, and work is still ongoing to look at the sequencing data from those. And again, watch this space. When we think about the GeCIP research projects across the whole of 100,000 genomes, there's over 245 projects which are registered, and the various GeCIPs then apply for external funding to support their research, and over £24 million has been awarded across all the different GeCIPs. So it's an enormous body of work, which again is still ongoing, but some of the first research papers in the more common cancer types, so bowel, lung and breast cancer, should be coming out quite soon. So the 100,000 Genomes project has wrapped up. The neuroendocrine tumour GeCIP's work is still ongoing, so we can provide updates when that evolves further. But at the end of the 100,000 Genomes project, there's an announcement by Matt Hancock, and this was before coronavirus, but there's an announcement about what the next 5 million genomes might look like when we sequence patients as part of the genomic medicine service within the NHS. And where cancer sits with this is, I think we will sit in this area here, where there'll be between another 500,000 and 1.5 million genomic tests. And that what we've learned from the 100,000 Genomes project is there's a few cancers where you do benefit by doing the whole genome sequencing, the whole 3 billion, but the majority of our more common cancers, there's a panel of 500 genes that you can look at to learn more about what might be the most appropriate treatment for those patients. But alongside that, there's going to be more sequencing of the UK Biobank, and there's also a real focus on looking at newborns and looking at areas of unmet needs, and also feeding sequencing into working alongside clinical trials. So this is what should be happening over the next few years, but as everyone is painfully aware, lots of things are up in the air with coronavirus at the moment. So just a quick summary of the 100,000 Genomes project. Genomic sequencing for all cancers will soon be the norm, and this can help tell us about the prognosis for patients. It can help us select the right treatments, and we can also learn about the pharmacogenomics, so toxicities of certain chemotherapy medications and other drugs as well. And looking at the legacy data from this work, we can see that 50% of cancers might have a mutation that would lead to either a new treatment or a clinical trial option. So what might be the opportunities for neuroendocrine tumours? Well, I've already discussed some of the germline, so inherited changes, so syndromes or other genes which might be inherited. Sometimes with neuroendocrine tumours, we don't know where the primary tumour has originated from, and that might be the case for some of you watching this today. And by looking at the DNA sequence in those tumours, it might help tell us where that primary tumour originated from, and that can be helpful for us in deciding the best treatment for people. We also know that looking at the DNA sequence can help improve the prognosis, so we can have more accurate discussions with patients about what we think the future might hold. And some of these changes might tell us how you might respond to your treatment that you're on. We also use it to decide what might be the best treatment and clinical trial selection. And also with the DPYD sequencing, we can understand about pharmacogenomics. And there are some things which we sort of label as tumour agnostic, because you might remember the slide about the tumour mutational burden. And sometimes that can give us an indication as to whether a patient might respond to immunotherapy, for example. But at this time, there isn't any routine use of immunotherapy in the more common neuroendocrine tumours. And then there's still work that's going on in terms of the neuroendocrine tumour GeCIP. So we're doing our best to understand as much as we can about the genomics of neuroendocrine tumours. So this is just a summary of the neuroendocrine tumour DNA sequencing work. What we've learned over the last 10 years is they have a very low background mutation rate. We know that the pancreatic and bronchial tumours have mutations in the epigenetic machinery in about 40% of cases. There's a number of syndromes and examples where germline mutations occur. We've learned that circulating tumour DNA and circulating tumour cells, they can correlate with outcome for patients, but these aren't routinely used at the moment. But what is challenging for us in neuroendocrine tumour research is we don't have these actionable mutations, and we don't have what we call hotspot mutations. Some cancers, there might just be one hotspot that you can target on to sequence to learn things, but we don't have these in neuroendocrine tumours. But it doesn't stop us from actually carrying on our research and doing as much as we can to understand the biology of these tumours. So I'd just like to thank you all very much for listening. But most importantly, I'd really like to thank all of the patients and their families for taking part in research and supporting us, because without your involvement, we wouldn't be able to do anything. So thank you very much.